Right, howdy. My name is Zachary Thompson-Aga, and I'll be doing a short presentation on quantitative PCR. So here's a brief over what I'm going to cover today. I'm going to cover what PCR is, okay, what quantitative PCR is, the fluorescent reporters they use in quantitative PCR, how to understand the results from quantitative PCR, the types of quantification, why is quantitative PCR important, and then just briefly we'll take a look at my works cited. So PCR, what is it? So essentially it's a technique in which you take the sample of DNA that you want to make copies of and you make a shit ton of copies of it. Okay? Um, Usually it occurs in cycles, okay? And each cycle has stages. But each cycle should double the amount of DNA that you have in your solution. Okay, theoretically. Okay. Now that's if you have everything you need for the cycle to work. Right, so you need your double-stranded DNA, right? Your sample. Okay, you need your DNTPs, you need a polymerase, and you need your primers. So in each cycle, there's three stages. Okay, there's a denaturing stage where it's heated up. The DNA is heated up to the point that denatures, leaving you with single-stranded DNA. Okay, it's cooled down a bit so that the primers can attach themselves to the single strands of DNA. And then it's heated back up a bit so that the polymerases can effectively do their thing. And now you should have double the amount of DNA, of the DNA that you want, theoretically. Okay. Now, some more on conventional PCR. It's qualitative, right? It's more of a, is that target there rather than quantitative. What I mean by that is that in conventional PCR, you don't know how many copies of DNA you've made after each cycle. All right. And the results from conventional PCR are typically analyzed through aqueous gel electrophoresis. Right? And what I mean by it's not exactly quantitative, it means that well, you can try to glean quantitative results from it. Like you can try to take how much DNA you've ended with and try to determine, I don't know, how much DNA you've made per cycle or how much you started with. But there's a lot of flaws, a lot of things that'll prevent that data from being accurate. Like you don't know if the polymerase actually faithfully made copies, right? You don't know if errors were made. You don't know if it actually made double the amount each cycle. Just a bunch of different things. Okay. Now, quantitative PCR is where it sort of fixes that issue. So it operates on the same principles of conventional PCR, right? The only major difference, right, is that it incorporates dyes and or probes. So what does that mean? So that means the amount of DNA and the fluorescence generated right during this whole setup is should be directly proportional. So measurements are taken at the end of each amplification cycle and it measures fluorescence. And theoretically, you should be able to determine how much DNA you generate after each cycle. Okay? Because it's related to the amount of fluorescence that's produced. And so you should somewhat get results. As you see in the graph below, more DNA is being generated and you get some graph somewhat like that. I'll explain that later. Okay, for now we're going to be covering fluorescent reporters. Okay, now there are two types. There's the dye-based, right? Something like SYBR green. And then there's probes. Okay, dyes work by displaying weak fluorescence in its unbound form and far greater fluorescence when it's bound to double-stranded DNA. Okay, probes generally work by having covalently attached fluorescent quencher that is released in the presence of a particular sequence. All right, now this is how it generally works. Now, in the picture that I show on the slide, it's sort of like two very, very common systems. I'll cover more. Okay, I'll talk more about it. Just take a quick look, right? Okay, let's move on. Dye-based quantitative PCR. Now, dyes aren't sequence specific. All right, what does that mean? It means that the fluorescence is directly proportional to the amount of double-stranded DNA. Right, so let's theoretically say you have contamination, you have junk DNA in there along with the stuff that you do want copies of. Well, it's gonna fluoresce regardless. As long as it's attached to double-stranded DNA, it's gonna fluoresce. So that's gonna affect your results. So it has reduced specificity, and you can only have one target. Right? You can only it's not specific. Right? Like I mentioned earlier, and it only requires 2 sequence-specific primers. So that's all the gist of it. Now, probes. Okay, probes are sequence specific. Okay, they must be designed specifically for each, let's say, gene or DNA that you want to copy. Okay, now it's increased cost and complexity. Okay, now there's something called multiplexing. Okay, it's essentially using multiple probes to target multiple different genes or sequences in a single experiment. Now, some say the limit is about 5, some say 4, some say 3. Just depends on what article you're reading and at what time the article was made. But many different types of probes exist. Okay. Now, I'm just going to cover a couple. Okay. First, one of the more common ones is called TechMan. I don't know if I'm pronouncing that right. Don't quote me on that. It essentially works. It's a sequence-specific, fluorescently labeled on it's a probe. Okay, it's a sequence-specific probe with a fluorescent reporter at one end. All right, and a quencher at the other. And essentially, what happens is that so it binds. Okay, during annealing and during extension, the probe is displaced, the border is cleaved, and the fluorescence, the free reporter fluoresces. Yeah, that's basically how that system works. The molecular beacon, okay, works somewhat the same. So it also is sequence specific, like pretty much all probes are. Um, so it binds during annealing, okay, and it separates the reporter and the quencher, and it allows the reporter to fluoresce. Right? So see this the pattern I mentioned earlier with basically all the probes. So here's a minor groove binder, and essentially it's attached to a minor groove binder, right? It's all tangled up, okay? And when it's all tangled up, the quencher and the reporter are close together so that the reporter doesn't fluoresce. And during annealing, the probe unfolds, right? The minor groove binder unfolds, and it uh, linearizes itself, and it separates the reporter and the quencher. Okay, another one, dual hybridization probes. All right, so you got your donor for it operates on FRET probes. Okay, so essentially during annealing, the two probes bind, okay, in a head-to-tail orientation, and then the reporter glows, right? So sequence specific. At the end of the day, probes are sequence specific. There's a lot of different mechanisms, and that's the entire gist of it. So before I go into understanding results, I need to throw out some definitions and other things at you. So quantitative PCR experiments should or typically do have 3 controls: no template control, negative control, and a positive control. Right? This is for the quantitative part of the experiment. I'll explain a bit more later. There's a baseline, the background fluorescent signal, basically. Um, RN, which is the fluorescent emission from the activated reporter compared to the emissions from the unactivated reporter. The negative RN, the RN value from the unreacted sample, RN plus the RN value from an assay with all the necessary things to have PCR go, and then you have a threshold, the average standard deviation of RN times an adjustable factor, CT, basically the cycle that generates enough emissions across the threshold. Okay, so here's something you typically get from a quantitative PCR experiment, right? This is a graph of a series of standards. Okay, this is a graph of a set of standards for a duplicate. The white line running through the graph is the threshold. The stuff you see at the bottom, right, is the background noise. Okay, now another thing is that you should know that it's the delta Rn, which is just basically the Rn plus um, minus the Rn minus. Okay, um, graph versus the cycle numbers. Okay, you should get you something like this, right? There's an exponential stage. It should be above the threshold, and then you have a linear phase where it kind of the PCR dies out. Okay, now you should also do a melting plot. Okay, which is done to determine the identity of the qPCR products, which is critical. Okay, knowing if you actually made copies of the right DNA sequence. And then let's talk about the types of quantification, right? There's absolute and then there's relative. Okay, the absolute requires standards, okay, to determine the target copy in an unknown sample, right? Same. For Here's a real life example correlating the viral copy number with a disease state. And in relative quantification, the CT values of the sample are compared to a reference. Okay, or for example, something like measuring gene expression in response to a drug. Okay, so those are the two types. So why is real-time PCR important? There's many different reasons. It's used for gene expression analysis, used for detection of mutations, particularly in cancer cells. It's used in food analysis. It's used in bioremediation monitoring. It's used in detection and quantification of pathogens. In fact, it was used extensively during COVID-19 for determining if someone had COVID. So here is my works cited, and thank you. Sorry about the bit of a stilted